Good day, everyone. Welcome to the Atchison Group and Clear Labs joint webinar titled From Chaos to Clarity, How the Next Generation Sequencing Empowers Food Safety. My name is Justin, and I'm responsible for scientific affairs at Clear Labs. And today we have three speakers with us on this webinar. So first up, we have Dr. Ben Miller, who has over 25 years of experience in food safety, public health, epidemiology, and infectious disease outbreak investigations. He currently serves as the Chief Operating Officer and Executive Vice President of Regulatory and Scientific Affairs at the Atchison Group, where he works across multiple sectors advising on food safety culture, traceability, public health, outbreak-related, and other scientific and regulatory issues. Ben has extensive food regulatory experience and as previous division director at the Minnesota Department of Agriculture, led the state of Minnesota's human and animal food regulatory work. In his role as the state regulator, Ben developed experience implementing legislative reforms on topics related to food safety and is an expert in FDA traceability requirements. He is also currently the vice chair of the International Association of Food Protection Program Committee and a founding member of the public-private FDA-sponsored Partnership for Food Traceability. Next, we have Dr. Dan Rice. He has over 30 years of experience in government sector, food safety, and public health programs spanning academia and regulatory science. Dan was the director of the New York State Food Lab for over a decade, and most recently served as the director of FDA's National Food Laboratory Program. He has substantial national and international experience in regulatory programs associated with human and animal food. Dan retired from the FDA in June 25 and joined TAG in July as a senior advisor for food safety. And finally, we have Carl Roden, who is the Director of Product at Clear Labs, where he leads the development of fully automated next-generation sequencing solutions to improve food safety, food manufacturing, and public health. He began his career as a food microbiologist and lab manager in Omaha, Nebraska, and later joined DuPont, supporting molecular diagnostic solutions for foodborne pathogen detection and characterization. Carl joined Clear Labs in 2018 and has over 20 years of experience in food safety and diagnostics, working to make genomics more accessible and impactful across the food industry and public health. So without further ado, I shall pass on the mic to Dr. Ben Miller, where he will take us through the webinar.
Thanks, Justin. Thanks, everybody. Great to be with you. I think hopefully this is a good, generate some good discussion within kind of your teams or even at the end of this webinar. But from at least my perspective, I think this is one of the bigger areas that we have some opportunities to kind of really lean in and use some emerging tools kind of within the laboratory and informatics space to solve some persistent food safety issues that we have, especially in food manufacturing. So, with that background, what are we trying to solve or what are we talking about today? And I think we've seen more of a movement now that people have become comfortable with developing some of their foundational environmental monitoring programs to how do they improve on those? How do they iterate those? And we're seeing again some options in the marketplace emerge that I think give people some tools to look at how can they do pattern clustering with genomics that don't create liability within kind of a regulatory or legal space, but allow them to generate or allow us as the food industry to generate some more actionable information as we think about root cause analysis and corrective action. So that's really the journey that I hope we go on together over the course of the next 45 minutes or so. But just wanted to start with that framing of kind of what is the question or what is the problem that we're trying to solve. And so anybody that's tried to really run an effective environmental monitoring program knows that it is very much a game of hide and go seek, right? Like, how do we determine what positives are related? Are they related? Did those positives come from an ingredient supplier? Did they come from a particular area in the facility? Do I have a strain that's persisting on biofilm or equipment? Or, well, another one comes in with raw material. So we're going to see kind of this diagram of this food facility a little bit later in the presentation when Kyle talks about the use of some of the clear labs technology. But I wanted to preface this with just saying, how are those red dots on that map related, and how do we tell? I think it's especially challenging when firms and companies are using just a positive, presumptive or absent kind of binary test, especially when we're looking just at Listeria species. And I think we've been cautious in terms of the way that we've wanted to access that information and not go beyond the Listeria species in a lot of instances, because we're afraid that we're going to create regulatory or legal liability if we're able to identify that we've got L mono in a facility. And so the mantra has been for probably the last 5 or 10 years, we'll just treat any Listeria species as if we were dealing with mono. And that is a strategy, but I think it also limits the allocation of resources in maybe a more or a less than efficient manner overall. And so, one of the takeaways I'd like you to think about as you watch this webinar today is, how might I better drive some of the root cause analysis that I have if I had more differentiating information? And what would that look like? Even if I didn't necessarily know that I had L mono, but I was able to actually see different strains within my environment. So kind of hold that thought as we go through this. I also kind of want to just propose that, I think this is an analogy that works for some people, but if we think about your EMP or our environmental monitoring program, what if we thought about it in the same way that we thought about our integrated pest management programs? Pests we can see, right? Like sometimes when I do food safety culture assessments, I give people a magic wand and I say, you get one wish, what would you use it on? Cast that spell. What's the problem you would solve? And they'd say, I'd want to be able to see bacteria. Well, that's really what we can do when we talk about environment or integrated pest management programs. How we deal with mice is different than we deal with flies, and so we're easily able to make that differentiation based on the visual evidence that we have in terms of controlling those particular hazards as they might be entering a facility. And so if we kind of extend that analogy, if we're testing at the genus level, it tells us that something's there, something's scurrying about. Maybe it's a mouse, maybe it's a rat, I don't know. Sequencing allows us to get more towards, is this the same mouse coming back or a new one getting in? And how we would deal with that is going to vary, right? In terms of, do we have a mouse nest in the facility? Do we have an infestation versus just something that's coming in because of, the weather's changing outside and we would need to tighten up doors and things like that. So if we're only testing at that level, we're not getting that sort of actionable level of information, much as we would, in terms of just managing pests at a maybe a simpler level within the facility. So maybe that's an analogy that resonates, maybe it's not, but it's one that helps me frame it up a little bit when we look at how we're trying to get more actionable information by doing some testing beyond just presence absent. And why is this important? I think anybody that's been in the food industry, and certainly over the last, 5-10 years, knows that regulatory agencies are using this type of information more to support the epidemiologic investigations that they have. So we've seen over the past several years a number of what I call slow moving outbreaks. These are typically related to Listeria mono, where there's just a handful of cases that occur, human cases of illness that occur over a year. Maybe there's even, like you can see in this epi curve here, a gap where we're not detecting many cases from like 2020 to the beginning of 2022, but it's persisting. And those regulatory and public health agencies are going to use that and continue to use that information from a whole genome sequencing standpoint to really drive their outbreak investigations. And that's going to include things like product testing and FDA and state-related swabathons, where they come in and obviously swab your facility and look for pathogens. And if they find something, they're going to go match that up to the NCBI isolate database and say, does this match any clinical cases or other food products that we've tested in the past? So the reality is that the public health and regulatory community is already using this technology and has been for quite a while. My colleague, Dr. Dan Rice, is going to talk about that kind of evolution or progression within how FDA has evolved from using kind of PFGE-based technology all the way to whole genome sequencing. And we're lucky to have Dan as part of TAG now as a senior advisor, coming off of his oversight of all the FDA food testing laboratories until about June of this year. So we'll have Dan kind of previewing what Dan's going to talk about in a few slides here. But I think the other thing to note is that there are shifting compliance expectations from the agency as well. So I just kind of highlighted some language from a couple warning letters, one from 2023, one from 2024. As you can see, that language is fairly consistent and almost identical. But in essence, we see the agency recommending the use of a technology like whole genome sequencing as a tool to investigate isolates from the environment. And again, why is that? It's really, if you read the language there, to drive that root cause analysis and corrective action. So the more information that we have about the isolates that we're detecting in a processing environment, post kill step, if there is a kill step and prior to packaging, the more effective we are to sort of muster or target the resources that we have, especially if you're finding a number of positives in your processing environment, which again, depending on what you're doing and what you're making, may be completely fine. Well-designed and EMP program finds positives. We want that to happen. The question is can we be more efficient in the way that we use that information once we find them? And I think it would also not be a complete presentation here if we didn't just talk about the March 2023 call to action letter that came out from FDA as well, where they really did lean into the infant formula manufacturing industry and say, following the issues that we saw around Chronobacter, that really using whole genome sequencing is a must for that high risk product category that's being produced. So again, just another indication that, from a public health and regulatory standpoint, we are seeing a movement there to start to leverage some of the things, some of these types of technologies. And so we're not going to overly focus on whole genome sequencing. When we get to Kyle's portion of the presentation, we're going to talk a little more about targeted next-gen sequencing, because I think that's a nice middle ground. I've got a few more slides on that. But before we do that, I wanted to invite Dan to the conversation and just have him give us a good background on where we've come over the last 20, 25, 30 years as it relates to the technology and how the agency has used it. So with that, Dan, I turn it over to you.
Okay, thanks, Ben. Want to talk a little bit about the history of genetic subtyping. The Food and Drug Administration's played a significant role in shifting from traditional typing methods into genetic-based typing methods over the years. And I've been fortunate to have been a lab person for quite a few decades now, and was working with foodborne pathogens before we really had any good typing tools that were based on genetic signals and attributes and so on. And so, so in the early days of genetic subtyping, we used pulse-field gel electrophoresis. And I was actually a graduate student many, many years ago using PFGE as a typing tool in the late 80s, early 90s, and participated in the Jack in the Box outbreak in 92, 93 in Washington State and Oregon, which I think really demonstrated the utility of PFGE as a typing tool for doing an epidemiological investigation. And so not long after that outbreak investigation, and where PFGE was used, I think really for the first time in a foodborne illness outbreak investigation, the CDC formed PulseNet in 1996, which used PFGE as a genetic fingerprinting tool for use in epidemiological investigations. And then PulseNet expanded to include local and state public health laboratories and also the FDA labs. Although PFGE was a great tool and it was demonstrated to be vastly superior to the tools that public health labs were using prior to PFGE, it still didn't have the resolution necessary to really pinpoint with 100% certainty the source of isolates and whether or not they were associated with human illness. So next slide, Justin. So embracing whole genome sequencing. So in 2012, FDA scientists demonstrated that WGS whole genome sequencing had superior resolution. They did a retrospective study of Salmonella isolates and outbreaks and were able to demonstrate that isolates that were identical by pulse field gel electrophoresis were at times actually very different genetically and not related. And so shortly after Genome Tracker was formed, which is the FDA platform that supports a number of local, state, tribal, territorial, and government labs that are focused on sequencing isolates from food and the environment and animal sources. And then these sequences are uploaded to a national, actually international database that's publicly accessible. And so the goal of the Genome Tracker program is to continually build the number of sequences in the database. And over time, it's really grown substantially. So the impact and application of WGS at FDA, it definitely enhanced outbreak investigations. I was actually, I joined FDA as a lab director. When I joined in 2015, we were still using PFG as our primary tool. Very shortly after, we transitioned over to WGS, and in my role as the national laboratory program, for most of that time, we were using WGS as our main typing tool, and it vastly improved our ability to identify outbreaks and investigate these outbreaks. As the state and local labs were transitioning over from PFG to WGS, so were the federal labs. So we have the PulseNet program, which was focused on the clinical isolates, and then Genome Tracker, which is really the food and environmental isolates that are coming into the public access database for doing the kinds of comparisons you would want to compare to see if isolates were related epidemiologically. So the FDA routinely uses WGS now for all of our outbreak investigations, and not only domestically, but also some global food supplies. Many of the investigations involve international perspectives. Next slide. So beyond foodborne pathogens, the Genome Tracker labs were in a great position to be used for more than just foodborne pathogens. So with COVID-19, these labs were doing wastewater analysis and sequencing for SARS-CoV. They still are doing this, not only just for SARS-CoV, but also they do this for influenza viruses, hepatitis viruses, using sequence analysis to demonstrate or to analyze similarity or differences of what's being recovered. And then, the FDA is more than just human and animal food. They've expanded the use of whole genome sequencing to other areas, such as the clinical validity of genetic tests and gene therapy development and so on. Next slide. All right, and so collaboration is key, and looking forward to the future. The FDA sequence program, Genome Tracker, is known to be highly collaborative and emphasizes standardizing procedures and platforms so that the data generated are very comparable. Routinely collaborate with other partners like NCBI, the National Center for Biotechnology Information. That's where the sequence data is all housed, and it's accessible by anyone that knows where to look. The USDA, the CDC, the World Health Organization, and the United Nations Food and Agriculture Organization. FDA, the regulatory entities, has had successful collaborations with industry using sequence analysis and sharing technologies and sequence data and so on, with a shared goal of improving food safety. And then the FDA, I just retired just just a month and a half ago or so. So when I left FDA, they were actively looking at other high resolution technologies to support foodborne illness investigations, including metagenomics and also next-gen sequencing, which is going to be a focus of what we'll be covering later in this webinar. So apologize for the technical difficulties, but hopefully you were able to follow my slides.
Thanks,
Pass
Dan.
off to Ben.
Yep, no, perfect. So I think when we look at where we're headed, as Dan talked about, the reality is that, from an industry standpoint, we need to start thinking about how we leverage some of these tools and really pick, I think, the right tool for the task. If you're a public health regulator, investigator, whole genome sequencing is really what you want to use. If you're a food company that's trying to figure out sort of what's happening in your processing environment, that may not be the right tool for the job. And I think that's what we're going to talk about here. There may be instances where it is, but where next-gen sequencing might really be the best option that you have, because at the end of the day, from a food industry standpoint, we need something that's fast, cost effective, and really provides that actionable information. So when we look at a table of, the kind of current state, where I'm going to say we test for Listeria species or Salmonella, and we're specifically looking for presence, absence, we're probably using PCR to figure out, what's there in the environment. Is that screen? We're not really getting any sort of genomic differentiation at that point. The cost is certainly low because it's the most prevalent technology right now, in the food testing space, and the time to results, it can be, quite good. There are some platforms out there that, have gotten down to, a number of hours, if you will, in terms of being able to say, am I dealing with Listeria or not? And the use case is really just where you're looking for presence absence within a particular processing environment. So it is the foundation or cornerstone of many of the EMP programs out there. I think what we're talking about in terms of progression is kind of, if we think about this crawl, walk, run, or good, better, best, kind of moving from that good to better in the space of targeted next-gen sequencing. And Kyle's going to pick up the thread here, at least through the Clear Labs perspective here in a minute. But where that, if we kind of look across some of those variables in terms of cost, the cost is still relatively low, and the time to result is quite good. It's still you're going to get a result within a time frame that you can definitely take action if you're trying to manage a processing environment. And when we think about the use case, it's really kind of in that day-to-day setting, right? It's driving and managing root cause investigation and corrective action follow-up. So if we go back to the beginning of what I was talking about, are we able to differentiate? Is this a resident? Is this a transient? How are we going to manage that? Are we seeing it, a particular strain come from a particular area of the facility, and are we seeing it move around? And so you're really able to take a much more tactical approach when you're trying to manage the limited time and resources that you have within that space. And I think this type of technology really allows companies to tackle that in a much more effective manner. Then finally, we've talked about whole genome sequencing. I think, we know that the resolution is quite high. We're able to say, are these exactly and absolutely related to each other? This sample versus this sample, if they're 20 SNPs difference or less, or 10, or however we want to define that technology can get us there. But it certainly is not necessarily as cost effective. And I think there are limited instances where food companies would want to use this type of approach on a regular basis when you think about managing your EMP program. So we're kind of in that sweet spot talking about targeted NGS for the purposes of this discussion. So with that, I believe we're going to hand the ball to Kyle, and you're going to kind of walk us through sort of what Clear Labs has developed within that space, and looking forward to hearing about it. So thank you. Kyle, it's yours.
Great. Thanks, Dr. Miller, Dr. Rice. That was a very nice presentation. I think you teed it up very nicely for us. So yeah, I'm going to spend some time talking about some of the real world applications and piggyback on a lot of what you guys had said already. Let me make sure that my screen's working here. Yep, looks like it. Okay, so So why are we talking about sequencing, right? Let's take a step back and just try to understand, why use it. And really, we're using it to get more information, right? We want to better understand the pathogen or the bug that we're encountering in our food or environment from the perspective of a food manufacturer. Enough information to make the right decisions, the best risk-based decisions. Um, and the reality is that there's a lot of technologies and methodologies out there that allow you to do that to an extent. For example, Salmonella serotyping or Listeria strain typing. Those tools exist, and they're all designed to, again, give you the most information possible to make risk-based decisions, monitor trends. Again, a lot of what the Ashton team was saying. Are you seeing an increase in the number of Salmonella serotypes or a particular strain over time? Are you able to pinpoint a particular organism to a supplier upstream? So you can go back to them and say, hey, we think that you're, consistently giving us this particular Salmonella or Listeria. And as they mentioned also, do I have a bug that is resident in my environment? Is this something I keep encountering over and over, which suggests that I keep cleaning, but I'm not getting rid of it? And at the end of the day, from a business perspective, a food manufacturing perspective, it's to give us kind of that operational, wherewithal to make the best decisions. So these methodologies, they exist out there today, but the question is, why isn't everybody using them? A lot of folks are using them, but they typically require, specialized laboratories. They're not necessarily cheap. They take quite a bit of time. And so they're not very accessible methods. You're dealing with live cultures. There's concern about, contaminating the laboratory and hopefully not contaminating, further locations. But there's a lot of reasons that folks aren't necessarily using them because by the time you get that result, a week has passed or maybe even 2 weeks where you've already made all your decisions. You've blasted the plant, you've made your corrective actions, and by the time you get that information back, it's more retrospective. So there's not really a whole lot of utility there, unfortunately. And we've been talking about, next generation sequencing, the value of sequencing. What are some of the values that can bring? But again, up until this point, this technology has remained pretty inaccessible. It's been very costly. You need even more specialized people to run it, and in the past, sometimes PhD level folks, executing these methods. There's always been a lot of concern about the level of information that I would be getting. Is this going to implicate me in some way? Is this data going to be misused or misinterpreted? The time to result historically has not been very quick. It's been, kind of along the lines of traditional serotyping or characterization where it's several days. You're typically sending this out to a laboratory and waiting several days to get the result back, at which point the data is, more retrospective in that sense. Has not really been easy to execute. And then along with like the level of information, the misuse of that information, there's legality concerns, right? There's regulatory perception. So there's a lot of reasons why folks have been reluctant to use sequencing technology. And what Clear Labs recognized was an opportunity here to kind of lower that bar to access sequencing and also make it safe to use, if that makes sense. We haven't really spent too much time talking about what sequencing is, and I won't dive into it here, but just to give you kind of an idea. So a lot of food manufacturers might rely on things like immunoassays or PCR assays. They're only looking for one target or maybe a small handful of targets. They amplify those and they detect them, and those are usually associated with the identity of that organism. That's worked pretty well, but what if you want more information? And that's really where sequencing comes in. So sequencing can do everything that PCR or immunoassays can in terms of detecting something, but it's looking at a lot more regions of the genome. So unlike PCR, where if you get one area of the genome that's positive or that hits, sequencing is looking for several, and so you need to check more boxes. And so along with that comes added assurance that yes, this is definitely Salmonella, because not only do I have one little area of the genome that looks like Salmonella, I've got several. So that's one really nice application of sequencing. And not to mention, if you wanted to take that even further, the areas that you're amplifying and then sequencing through, if you wanted to, you could detect things like, is it resistant to antimicrobials? Are there different properties, virulence factors that I might be interested in? I could sequence through those and actually get that information. So those are some of the critical differences between traditional molecular methods and sequencing. So at Clear Labs, again, we saw an opportunity to make this technology more accessible, safer to use, and we have this platform called Clear Safety. And it's very, very easy to use in terms of taking what you've already enriched, typically with food samples or environmental samples, you enrich those, right? And then you put them on a PCR or a lateral flow strip or whatever it may be. The process is pretty much the same. You take that enrichment, put it on our platform along with the necessary reagents, consumables, and the appropriate kit, and then you get your interpreted results. So it's less than 30 minutes of hands-on time. It's very, very easy to use. We have a lot of customers where, folks have, been working at that company for a while, maybe on the production floor, and they get promoted to the laboratory. They don't necessarily know much about sequencing or how to operate these kind of things, but it's so intuitive that we have folks they're using this on a routine basis, with minimal training and experience. I want to mention that, despite our name Clear Labs, we're not necessarily a service laboratory providing this as a service. These are actually units that are deployed to customers at food manufacturing plants, at service laboratories, at public health laboratories. So we provide the platform, the reagents, the consumables, the installation, the training, the software, the support, and the user deals with Clear Labs. So it's a system that we deploy to various laboratories across the world now. We have two different platforms. I'm going to tee up kind of the conversation about targeted NGS, like the Ashton team mentioned, and whole genome. So the platform that we offer to food manufacturers is Clear Safety, and we have solutions for Salmonella and Listeria detection and characterization. The time to result is very competitive with what food manufacturers might experience today at just the detection level. So looking at next day results from the enrichment. And importantly, this is only analyzing a fraction of the genome. It is not analyzing the entire or the whole genome. So in other words, you don't get this SNP or single nucleotide polymorphism resolution that you might get with whole genome. We do have a whole genome platform on the right here. This is called ClearDX, and it's actually in use by a lot of the public health laboratories and federal laboratories in the United States. That will do a whole genome. And really, I think what we recognize and understand is that the needs of the food industry are different than the needs of the regulators, the public health. They have the same goal, right? But they have different requirements for the data that they need to make the best decisions. So I'll talk briefly at the end about our ClearDX solution. And like the Atchison team said, there are applications for food manufacturers who use this type of technology. But I think what we want to try to get home here is that really a targeted kind of middle of the road approach is probably the most practical. And I'm going to attempt to illustrate this here. This is kind of similar to the analogy that Dr. Miller mentioned earlier with the mice. So with targeted, like I said, you're not getting the complete picture. You're getting enough information to be able to see what the problem is. So in this case, I've got a picture of a dog, and they're puzzles, right? And on the left, this is representing targeted, where I'm getting a lot of the pieces of the puzzle, and then it's enough to make out what it is, right? And so I can know immediately right away. I think that's like a King Charles Spaniel, but I know enough to make the decisions from a practical food manufacturing perspective. On the right, you have whole genome, where you're getting every single piece of the puzzle. And that's something that regulators, CDC, FDA, USDA need in order to conclusively, legally say, yep, this is 100% certain our issue. Food manufacturers don't necessarily need that. They need enough information to kind of keep themselves from getting to the point where FDA, USDA is coming back and saying, hey, we have whole genome. We see that you have a problem that you haven't been addressing. So hopefully that analogy works here. So I mentioned earlier, we have two solutions, Clear Safety Salmonella and Clear Safety Listeria. You can think of these as kits. So with food manufacturing, food testing diagnostics, you have like a kit for Salmonella, Listeria, that's either PCR based or immunoassay. It's the same concept here. So Clear Safety Salmonella has primers built into this kit that are specific to Salmonella. Listeria has primers specific to Listeria. But at the end of the day, what they both are rapid, fully automated next-gen sequencing solutions. As I mentioned earlier, you put the enrichment on the platform, you put the respective kit on the platform. The system does all the on-deck lysis, dead cell mitigation, so it won't pick up any dead cells, which is a problem that's experienced with other testing methodologies. And what the really neat thing about this is that we have what's called tunable characterization. So in other words, depending on the amount of information I want about that sample, I can tell the system, I want to know, is it this species or not? Do I just want to know genus level? I want to know, is it this serotype of Salmonella, or is it only one serotype? Is it dozens of different serotypes? For Listeria, do I want to know, is it the same strain I've been encountering before or not? So that flexibility is built in there because different manufacturers have different categories of food and risk, and they might want different amounts of information with respect to one another. So for Salmonella, we can report it's actually close to 70 serotypes now. And importantly, for both assays, we can determine co-infection, which means if you've got more than one serotype or species in that sample, NGS will pick it up, which is not something that traditional assays, unless you have a multiplex PCR that you can do. It's fairly high throughput. You can get close to 200 samples in a batch if you wanted to. And again, the time to result, 12 hours from enrichment, which is pretty competitive compared to traditional methodologies. You're getting that much more information a lot faster than you would be if you were sending it out to a specialized laboratory, or if you're performing those kind of traditional methodologies where it's taking you several days, several hours, even weeks in some cases. So it's a lot more actionable, if that makes sense. On the Listeria side, I got some cool screenshots coming up where it's really helpful to put NGS data to work for you and to visualize this information on, say, an environmental map. So I've got some examples of that as well. But the concept there is that you don't have to decipher this information. We're putting it to work for you, if that makes sense. Okay, hopefully I didn't click twice there. This concept of tunable resolution, I apologize, the screen is a little bit small, but when you're registering a sample in our platform, like I mentioned, there's an option for Salmonella and Listeria. For Salmonella, if you just want to know if you have a particular serotype in that sample, you can select that serotype. Or if you want to know if you have any of these serotypes in your sample, you can select all of them. So that's what we call tunable resolution. On the Listeria side, since the needs and kind of the understanding, the thinking around Listeria is different. So in Listeria, we're more interested in maybe what the species are, maybe especially El Mono. But there's also this growing body of evidence that suggests that these other species kind of run in the same circles as mono. So if you do want to know if you've got these other species, you can test for that. Alternatively, you can uncheck all those. And if you just want to know if you have genus Listeria, you can get that information as well. But the cool thing there is that even if you just want to know if you have genus, you can do this option called similarity analysis, where it will test for genus Listeria, but it'll also tell you after you get the result, have you seen this particular Listeria before? Even though you're not going to necessarily the species level or reporting that species level, it'll just tell you, yep, you've got Listeria and you've seen this particular Listeria in the past, because our software, as I'll show you, kind of banks those and creates these groups or databases based on what you've encountered historically. Showing you what the results are with this concept of tunable resolution. So in this case on Salmonella, on the left, we do see that it was positive for Salmonella enterica. And by the way, it was Salmonella typhimurium that was detected. In some cases, you might have a couple serotypes listed here, or depending on what you select upstream, like I mentioned. On Listeria, you can see we do have a positive sample here for Listeria, but we didn't test specifically for any of the species. So we don't have that information. We just know this Listeria. But we've also got this similarity analysis that we enabled during that registration. We do see there's a pattern detected here. I've got this, we assign this kind of identifier, this number, we call a pattern group. And within this group are strains that you've encountered historically that our system determines are the same. And so that kind of lets you know if you've got a persistent problem without necessarily knowing is it mono or not. Because in a lot of cases, you're going to treat Listeria the same anyway, but you don't necessarily have that mono tag associated with it. One of the other really neat things about NGS that I've been talking about is this ability to do co-infection determination. So with traditional methods where you culture out your positive sample, you get some colonies on a plate, and then you might take those on, or you might send that to a laboratory and they might take them on for serology or downstream strain typing. This becomes kind of a sampling bias game, because you've got a number of different colonies on a plate. You don't necessarily know if they're all the same. You're probably going to pick a couple and hope that you get a good representation there, but then you run that downstream and you find that you only got one serotype or one strain. What we've demonstrated is that if you have a plate, which could very likely, and this happens in the real world all the time, that is co-infected, you've got a few different strains of Salmonella or Listeria on there. NGS doesn't care which ones you pick, because it's going to detect all those strains in the enrichment that you tested already on the platform. So that's actually opening up a lot of eyes from our customers that have adopted us over the last 8 years or so, where they didn't have that available. They didn't know that their samples actually had a mix culture of maybe a couple different species of Listeria or a couple different serotypes of Salmonella. We have contracted a couple of blinded studies through a service laboratory where they have our platform, and we send them what we know are pure Salmonella serotypes, some single infection, some dual infection, send those blinded to the lab, say, just run these. We know what they are. They're ATC strains. And we see, do they get the expected results? And we ask them to compare it to their whatever they offer for their traditional serotyping. And they do that. What we found is that we met the mark every time. We detected what we expected on clear safety, but with the competing platform, they didn't quite match up. They didn't get all 7 of these serotype calls like we did. So that's just demonstrating some of the deficiencies in traditional methodologies. Then what about when you inoculate with 2 serotypes of Salmonella? Well, the same outcome was observed with clear safety. We detected both serotypes in each of those samples. And in this case, the competing platform did even worse because again, it's a sampling game, it's a numbers game. You're kind of relying on making sure you pick enough colonies and take those to downstream methods. And in this case, they did pretty poorly. So that's why a lot of our customers have been surprised that wow, we didn't necessarily know that some of our samples have mixed colonies in these enrichments. So it's been quite the eye opener. So this is kind of a nice illustration about, like I mentioned earlier, putting your NGS data to work. So we came out with this concept of environmental mapping, in particular for the Listeria product. And what a lot of food manufacturers do today is they might do some sort of mapping, but really they're just a bunch of red dots on a floor plan, right? You don't really know what to do. Should I just blast the entire place, right? It's kind of inefficient, like Dr. Miller was saying earlier. It's all about operational efficiencies. I could blast the entire place, shut everything down for a week, clean everything, but do I really need to do that? Am I really addressing the source of the problem? Well, if I start doing strain typing using something like targeted NGS, I can see some interesting things here. So I can see that I've got 3 different strains of Listeria, this purple group here, this green group here, and then this kind of red loner out here. And then what I can do is start adding some metadata. When were these sampled? What day, time? I can start to draw some conclusions. And okay, these guys are associated with one another. Maybe I can even see how they're, based on the time window, how they're moving around. So there's a lot of neat things that I can do. And it's all about really understanding the problem, not just knowing that you have a Listeria problem. It's really understanding it, which I think is really, really critical to properly addressing it and getting to the root cause and making sure it doesn't occur again in the future. And so I'm going to go back to that screenshot that Dr. Miller showed earlier. And this is a real world example with our software. Users can upload like a CAD or a PDF image of their floor plan, and they can start to see the results. And they can see a bunch of red dots, but the neat thing with ours is I talked about this concept of pattern grouping where they can start to see these similarities. And in this case, this is just one group selected. So these several Listeria strains are all related. They're all the same. And what this allowed the manufacturer to do, they said, well, why do I have these guys way over here and this one loner over here? How are they getting back and forth? How did this strain get shared over there? And what they found is that there's actually an irrigation pipeline that runs from this guy in the lower right here all the way over to the top left. And they identified that as the route of transmission. And so they were able to go back to this area, do a little bit more in-depth sampling and identify more of this strain. And that was really the root cause of their problem. And so you can imagine they could have spent months, years even, dealing with this problem and not necessarily addressing the root cause. And this type of technology helped them do it in a couple of days. So pretty powerful stuff. Talking more about Listeria. So we kind of did the same exercise as we did for Salmonella, where we sent samples to a laboratory that has clear safety. They were blinded, and we want to see for our Listeria strain typing or pattern grouping, can it correctly put the same Listeria into groups, and can it also properly exclude Listeria that are not the same? And we use WGS as the comparator. Like we said, WGS is kind of the gold standard. That's going to tell you with certainty that these are the same strains. And we asked the laboratory to use their traditional Listeria strain typing technology, which is a molecular-based technology. And what we found is that with clear safety, we sent 2 different batches, a smaller batch and a bigger batch, all different strains. But we knew which ones should be the same. We sent replicates and also which ones were different. And what we found is that clear safety matched whole genome every time. The competing typing platform failed to properly group Listeria that should have been the same or should have been grouped together in both cases. And we did a similar exercise where we wanted to make sure that clear safety was not grouping Listeria that weren't the same, right? So we have 5 different Listeria, they should be 5 different strains. So clear safety, sure enough, said that, yeah, these are all different strains. They should not be grouped together. Whereas the competing molecular-based typing platform was incorrectly grouping Listeria together that WGS said were not the same. So a lot of those technologies that have been out there for the last several years, we've been able to demonstrate that they don't necessarily have the utility or the value that something like targeted NGS does. So I'm showing this slide again because now I want to talk just briefly about our ClearDX platform, which I mentioned is in use in more public health laboratories, but that's not to say that food manufacturers can't use it as well. The applications are probably different. You might be using it more for in-depth investigations. You might be using it to rule yourself out of what might be believed to be an outbreak that's associated with your food, your environment. They might be using it for more R&D purposes, strain integrity, intellectual property, those kind of things. So I just wanted to mention that we do have a platform that exists compared to traditional whole genome sequencing methods that are out there. It's really the same song I was singing earlier. It's neat, but if you want to have whole genome sequencing perform on something, you have to send it out to a laboratory. It takes several days. Or if you have your own centralized laboratory, there's nothing really automated. It's very manual. You need specialized, personnel executing it, interpreting the data. We saw an opportunity there, and it was really at the height of the pandemic, but this is a platform that put a sample in, and next day you get a whole genome sequence. 30 minutes of hands-on time. That's kind of our bread and butter. The platform is so easy to use, anybody can use it. And by the way, it gets you these results that are actionable, within a time frame that you can actually do something about it. Compared to these legacy methods that are very time-consuming, a lot of hands-on time, a lot of opportunities for error. That solution exists as well. I just wanted to make that apparent. So in conclusion, like the Ashton team was saying, kind of a middle ground targeted sequencing approach is really the more practical, cost-effective solution for monitoring. You're not necessarily getting all the information that somebody might need to legally implicate an illness with a particular strain. It's giving you enough information to make the best risk-based decisions efficiently. WGS probably not needed day to day or for routine, but it does have its applications, like I mentioned, for more research and development purposes, more in-depth investigations, things like that. So I think that's my last slide, and I believe I'll turn it back over to you, Ben, to wrap up.
Absolutely. Thank you, Kyle. Yeah, that was, one of the things that stands out to me, I think, just in terms of the kind of the platform that you've developed, is that ability to sort of dial the level of differentiation. Do I want to know it's Listeria? Do I want to know it's mono? I think that really is a powerful tool as we talk about just the application of why somebody would want to think about this. So, I think from our perspective and working with clients that have implemented some kind of NGS or targeted NGS, we have seen there is a value proposition there, especially in terms of that root cause investigation and corrective action. So the ability to find some of those niche environments more quickly, right? Like all of a sudden I've got this piece of equipment and everything seems to be coming from there. I think intuitively we try to get there in terms of just kind of presence absence testing, but the pattern grouping that Kyle was talking about really allows you to kind of focus in and say, okay, well, maybe I've got two niches, right? And how I want to address those may be different once I'm able to nail down exactly what the source of that looks like. And likewise, when something does appear to be new, it does allow that kind of focused resource allocation, right? I've got my resident strain. I know I'm kind of, driving towards a complete corrective action around that resident strain. But if there's something that's new entering the environment, is that coming in ingredients? Does that have the potential of becoming a resident strain? And how do I really kind of focus my firepower, if you will, in terms of something that might be kind of new popping up on the radar? So again, I think just more information is more useful when you're trying to really manage an effective EMP program. All right. And so kind of just leaving attendees with kind of that, what should I do next? I think it's always good to review your EMP program, regardless of what method you're using. So, we like to do this when we go in with clients and do, a food safety gap assessment. The EMP is one of the kind of cornerstone programs that we'll look at if you're making a ready-to-eat food. So are you swabbing in the right places? Are you doing it often enough? Are you calculating and tracking your overall positive rates, by area or by line or by function? What do those look like? How have those changed over time? Is there any seasonality? Does that drive, the number of swabs that you're taking? So obviously we're talking about a technology to analyze the results that you get, but much of it is really informed by how you're actually collecting those results, and are you getting enough from the right places at the right frequency to take appropriate corrective action. And then kind of that next piece is your root cause analysis program and process strong, and once you find the root cause of something, do you have good corrective action plans in place. I hate to say, but I think sometimes we see companies really, they're focused on the cleaning and sanitation piece of it. Yep, we found a positive, we go blasted it, like we did our vector swabbing, we didn't find anything. And then a week or two or a month later, you're back where you're finding positives in that same area of the plant or same piece of equipment. And so there you're sort of treating the problem, you're not necessarily solving the problem. And again, getting this more kind of pattern grouping information can really help drive that. I think after you've done those two things, it's a great time to have a discussion with your food safety team where you can say, is this type of analysis, can it add value to our programs and help us, really kind of target the resources time that we have to reduce risk in our processing environment. So again, I'm always going to encourage people to look at their foundational programs, make sure that those are strong, and then lean into a technological solution. But I think once you're in that place, the information that you can gain by using something like TNGS, there's a lot of value there potentially. I always like to end with a quote, or at least usually end with a quote. So this one stuck out to me in the context of this discussion. It comes from Aldous Huxley, who wrote Brave New World. If you haven't read it, it is worth a read. It's a little bit of a strange book, but he was an author around the beginning of the 1900s. Said, facts do not cease to exist because they are ignored. So not knowing what we're dealing with doesn't change the fact that that reality is there. And so, using the information, going and seeking more facts can really help us drive corrective actions and ultimately lead to the fact that we're able to produce food more safely and more confidently. So with that, we're almost at time, but I appreciate everybody that had a chance to attend, and thank you to my co-presenters. It was a good discussion, and I think we've got time for a few questions if there are. Thank you.
Well, thank you, speakers. And as Dr. Miller has mentioned, we are coming up to an hour. We do have a couple of questions, but unfortunately, due to the time limit, we will be addressing them directly back to you all via email. So thank you all attendees for joining us. You can reach out to us, be it Clear Labs or the TAG group, via our email, our website, or the phone number as published here. So just a heads up, this webinar was recorded and you can watch it on demand as and when you would like to review the information. Feel free to share it with your colleagues, your collaborators, or anyone you think would be interested in the topic that we have touched on today. So, well, without further ado, thank you once again, speakers, for such an insightful webinar. And with that, we would like to close the webinar. Thank you all and have a lovely week ahead.